Good morning, dear students. Today we'll discuss Physiology of Synapse, Competency PY 10.2 as per NMC. So in the previous lecture, we have seen this brief sketch of working of central nervous system. Basically, the response of central nervous system is either in response to sensory stimulation, like stimulation coming from this receptor, which will travel from sensory nerve ascending tract to sensory cortex, or it is in response to an idea in the mind, just like I want to write on this board with your chalk. So this is the idea. So for now there will be motor action which will start generating in the cortical association area, premotor cortex and motor cortex. So by this combination of all this, the motor orders are formed, but it will not go directly. It will take opinion from two its siblings. One is basal ganglion and other is a cerebellum. That's why these two are called as motor coordination area. Now from here impulse will come back again to motor cortex, and it will come down through descending tract, either directly or indirectly by taking station in between, called as brainstem. Ultimately it will go down to main executing organ, that is a muscle or a gland, through this nerve. So today we will discuss basically, in the previous lecture we have discussed regarding receptor. We now will discuss regarding synapse. So to start with, objectives of towards today's lectures are: what is the synapse, types, that is anatomical and physiological synapse, physiologic anatomy of chemical synapse, which is the most common one, and postsynaptic potentials, neurotransmitters. Okay, just to have a recap, basically nervous system is nothing but bunch of neurons having cell body, axon and dendrites, and two neurons connected with each other by a junction called as synapse. So basically CNS is nothing but bunch of neurons and there are synapses. So to have a close look on synapse, this is the cell body, dendrites, axons, and these are axon branches, other neurons, axon terminals. Now anatomically, the synapses are basically the axons of one are ending either on the dendrites of other axon, just like this. So it will be called as exodendritic synapse, or they are ending on the cell body, it is called as exosomatic, or they are ending on some axons, so it's called as exoaxonal. So like this, junctions where axons or some portion of one neuron terminate on the dendrites or soma or axon of other neuron. So anatomical types are basically three: exoaxonic, exosomatic and exodendritic synapse. So here some of the examples I'm showing where are the synapses in the nervous system. Everywhere you can see here the impulse arising from the receptor going back to the muscle. So they can be, or it can go to a segment above, that is from T10 to T9, or like this, or some impulse are going to the higher, that is through ascending tract. So we'll have a synapse. You can see here at this position we can have a synapse. We'll have a synapse here. So like this, at multiple places there is a communication between neurons. Here you can see in the ascending tract, at all places you will have a junctions here. So these are the areas where there is a modification of the responses basically. So these are the locations of synapse in the nervous system. Similarly, for the descending tracts, impulse coming down from up to down. While coming down, so many junctions are there. Okay. So each neuron basically divides to form over 2000 synaptic endings. Human nervous system having 10 raised to 11 neurons. There are about 2 into 10 raised to 14 synapses. Just imagine how many synaptic connections. Synapses are dynamic structures, increasing and decreasing in complexity and number with use and experience. So here comes the concept of if you don't use it, lose it. And if you use it, it will be going on better development. So basically the concept of memory is nothing but based on synaptic connections only. So to one thing we repeat so many times, synapse develops and we remember that thing. Other thing we don't repeat, synaptic connections become poor and forget it. So basically memory is nothing but it's a physiologically synaptic development, either in the positive way or a negative way. Physiologically, there are two types of synapses. One is called as a chemical synapse, which is the most common type. Other is electrical synapse, which are at some places of nervous system. Electrical synapse is basically just like gap junctions, just like we are having sensitium in the cardiac muscle. If you stimulate a single cardiac muscle, immediately it will spread to whole through a gap junctions, through that ions transfer from one to other. Similarly, in the neurons, there are gap junctions that allows free movement of ions from interior of one cell to interior of the next cell. So, but it is at very few places. Most common we get are the chemical synapse. The working is same as you have seen in the NN junction, that is neuromuscular junction in the muscle. A presynaptic terminal, soma or cell body of other neuron. This is the synaptic cleft. Transmitter vesicles are there, which consist of the neurotransmitters which are formed in the cell body, travel along the axon and reaching to the axon terminal here. So these are the main things which are helping in deciding what type of response will be there in the postsynaptic terminal. Receptor proteins are there. Here these are again very important. They are leading to modification of the responses. Many times neurotransmitter is same, but response changes based on the receptor proteins, and this is the great area of modification of synaptic potentials. Close look to this one. So these are the synaptic vesicles which are normally formed in the cell body, travel along like this and they reach the terminal. So this synaptic vesicle consists of the neurotransmitters. This is the axon terminal, dendrite, postsynaptic neuron. This is synaptic cleft. So just have a close look again. These are the microtubules present at the endings of the presynaptic terminals which are helping in the movement. Mitochondria. These are the clear vesicles, active zone and postsynaptic density. So postsynaptic density, specific receptors, binding proteins, enzymes. These are all the area which are very, very crucial. And here is all our nervous system, medicine, nervous system modification works on this one. So it's a great area of research. If you want to do, if you want to promote some action of synapse, if you want to demote it, everything, ultimately you can do the modification at this place, induced by postsynaptic effects. So just seeing the sequential events across the synapse. Means when you stimulate one neuron, it will stimulate other neuron or to inhibit it. The cascade of event is same just like at the neuromuscular junction. First, whenever the impulse is arising like this, action potential arising at the terminal. First event is again voltage gated calcium channels are open, influx of calcium. Now this will take the vesicles near the site for the dispatch for exocytosis. Now the neurotransmitters are released. Now the neurotransmitter will combine with postsynaptic receptors. So these are the ligand gated channels open, and there is generation of action potential in the next neuron, that is called as a postsynaptic potential. Simple events are there. leading to nerve impulse. So one neuron will stimulate the other neuron by this cascade of events. So just flow of events. Action potential reaches the terminal, depolarizes the presynaptic membrane, leading to opening of calcium channels, influx of calcium ions, the transmitter substance released at synaptic ending. Transmitter binds to postsynaptic neuron receptor. Postsynaptic potential develops. Now this potential develops is basically of two types, either inhibition or excitation. So nervous system is very simple. Basically it is only three: neuron, synapse and synaptic potential, which either inhibition or excitation. In this three only we will be working on nervous system. Whether you take a basal ganglion, cerebellum, spinal cord, cortex, everywhere, this is only just like a computer. Three things are there only: neurons, synapse and synaptic potential. And we have to work on synaptic potentials only. If you want to increase activity of some neuron, we have to give we have to work on the postsynaptic potentials only. So close look at this terminal release. Synaptic vesicle, postsynaptic membrane. This is the neurotransmitter which are released, and this is the synaptic cleft. This is the close look of the postsynaptic proteins which are present. So neurotransmitters combine, here subsequent event takes place. So when the neurotransmitter combines, there will be opening of some channels and closing of some channels. So if there is opening of sodium channel, there will be influx of sodium leading to depolarization. That is what we call as excitation. So this is called as, so excitation is by influx of sodium, just like at the muscle we have seen at so many places. So this is excitatory postsynaptic potential. So it depends on again the neuron and the receptor protein. Neurotransmitter and the receptor protein. So if the neurotransmitter release is excitatory, it will combine with the receptor proteins, which again has to be excitatory, and there is opening of channels. Sometimes transmitter is same, but the receptor proteins are of different types. We will see how the difference takes place. So this is the typical action potential generation. If you have given a small stimulation to 4 neurons, 4 synapses will fire, but they might not be able to generate action potential in subsequent neuron here. So it will not develop action potential. If you stimulate more, some more neurons are excited, but if you give a threshold stimulus for that neuron, more neurons will be excited, just like here, for example, 16 neurons are able to fire. So like this, potential for the different neurons are different. Ultimately, action potential generate which travel along the postsynaptic membrane. And if the neurotransmitter release is inhibitory one, just like in GABA, glycine, it will lead to opening of potassium channel. There will be efflux of potassium or influx of chloride. So this will lead to hyperpolarization. So that is inhibition. So the neuron is inhibited. So this potential is called as inhibitory postsynaptic potential. So these are the only two. One is excitation, one is inhibition. So postsynaptic potentials are either excitatory or it is inhibitory. But again, inhibitory, there are so many types of inhibitions are there. Now the neurotransmitters which are common, we have discovered up till now: acetylcholine, catecholamines, dopamine, GABA, glycine, and few more are there. But see, always remember, these are the things which we know up till now. Maybe in the future we'll have so many transmitters, because still in the nervous system we don't have good answer to so many diseases. For example, basal ganglion, we are yet to cure it. So many neuronic disorders are there which we don't have answer at present. We are just able to control them, but we are not able to cure it. Maybe in the future, scientists from your batch or other one maybe find some neurotransmitter which can be blocked or which can be promoted, and due to that we can find an answer to some diseases. So still go in the nervous system with a positive mind that these are the things which we know up till now. Maybe in the future some new things will come. Basically, actions of transmitter depends on the fate of the neurotransmitter. Sometimes the neurotransmitter diffuses out of the synaptic cleft and action finishes. Sometimes enzymatic degradation, for example, acetylcholine esterase is present which degrades acetylcholine and the action stops. So here again we can work on that one. If you are giving this blocker of acetylcholine esterase, action will remain for more period of time. Reuptake into the cell, for example in case of norepinephrine, dopamine, or sometimes metabolized by monoaminooxidase inhibitor. So these are the areas where we can work, we can block this, we can promote this. So like this, if you want to increase action of the neurotransmitter, we can block its destroyer, or we can reduce the uptake of this also. So these are the areas where we can work and have the action at the synaptic endings. So these are the cascade of events which we have discussed. Impulse which will lead to opening of voltage gated calcium channels, influx. The vesicle comes near and there is a release of neurotransmitter, ultimately fate of transmitter. If the transmitter remains for more period of time, potential development will be more. If the transmitter is removed immediately, the duration will be reduced. So this will lead to generation of action potential. So these are the different types of proteins present at the synaptic membrane. So intravenous toxin, it acts on septobrevins. Apart from neurotransmitter, there are so many substances which also act on the transmitter, and either they block the action or they promote it. So like commonly the tetanus toxin, it is acting on synaptobrevin, which causes spastic paralysis by blocking presynaptic transmitters released in the CNS. Botulinum toxin C acts on the syntaxin. Botulism causes flaccid paralysis by blocking the release of acetylcholine at neuromuscular junction. So like this, information regarding the synapse will help us understand so many disorders, because ultimately if any neurotoxin or any external body is acting on the neuron, it will be acting only on synapse, which will modify either the stimulation or inhibition, which will again lead to the actions. Local injection of small dose of botulinum toxin is used in treatment of muscle hyperactivity. For example, it's muscle hyperactivity. We use this to relax the muscle. So here we are finished the basic synapse. We have seen that the neurons are there which are connected to each other by synaptic connection, and synapse actions are only two, either excitation or inhibition. So in next lecture we'll learn in detail regarding properties of synapse. Till then, thank you.